Some other jobs I've had, I was kind of like just using box. Box. Hey, how are you?
Hey. Hold on a minute. All right, hello, can you hear me?
Yep.
Oh. Get you through the earbuds.
Yeah, do you see this?
Yeah, I got it. Okay, good. Oh, yeah. That's funny.
Yeah. All right. So, do you have any PowerPoints to send me?
Yes, I'm actually, that's what I was in the process of getting really quick. And I have a sample test as well. We can meet tomorrow too, yeah?
Yeah.
Could we do 12 to 3 tomorrow? Is that possible?
Yeah, we could do that.
All right, good. Because at 3 o'clock I have a therapy appointment.
Okay.
And then you have the membrane transport one, or is that the, you have membrane structure? Which one do you have?
Let me see.
I know we're almost finished with one of them.
Yeah, I do.
All right, so I'm sending you the stuff now. And there's 2 outlines there that some students made that we shared along. So if you see anything worth going over on one of those, we can do that. And where's membrane structure? Here we go. We're on slide 55 or 67. Okay. Did you get the email?
Let's see. Yeah.
So they got closer to finding out what I have. Remember?
Yeah?
Yeah, they got closer.
What are they?
I'm going for a colonoscopy on the 18th and stuff, but it's coming down to Crohn's disease.
Oh, really?
Yes,
Yeah.
sadly so. But also happily so, because then I won't be in pain for too much longer.
Say that last part again?
Huh? No, I said that it's good because I won't be in pain for that much longer, hopefully. Because then they'll give me steroids and I just take a medication.
Mm-hmm.
Even gotten to the point where they have given me medical marijuana.
Really?
Yeah. The doctor recommended me for it and all. And I have my certification from the state and everything.
Really?
Yep. Quite crazy, but it happened.
Mm-hmm.
I was very surprised. All right. We're on slide 55 over here.
On the
Or
membrane
54. How do we study protein dynamics in the plasma membrane?
one? Okay. Let's see. Protein movements, diffusion of membrane proteins after cell fusion.
Yeah.
Okay, let's see. A mouse cell, rhodamine labeled membrane protein, and then fluorescin labeled membrane protein. Okay, in a human cell, they fuse the cells, and so you have half like the rhodamine labeled stuff and half the fluorescin labeled stuff at time equals 0 minutes after cell fusion, and then after you incubate it at 37 degrees Celsius, 40 minutes later you have a mosaic. So that's really just, it's kind of obvious. Let's see. Transition temperature 59 degrees Fahrenheit. So that's kind of a graph that measures the mosaic percentage on the y-axis and incubation temperature on the x-axis, and apparently at 59 degrees Fahrenheit, or that seems to be like around maybe, I don't know, 16, 17 degrees Celsius, then it goes like flips over to the mosaic kind of diffusion.
Got it. Okay.
And then there's FRAP, which stands for Fluorescence Recovery After Photo Bleaching. So we essentially have some type of like, all right, so I see like membrane proteins here. The green things are the membrane proteins, I believe. And there's fluorescent tags attached to them. That's what the kind of sun, kind of looks like a sun.
Yeah.
All right, and then they bleach it with a laser beam. So they basically, I suppose they remove the fluorescent tags in some area, and they call that bleaching.
Okay.
So that area is going to appear white instead of like green, I suppose. And that corresponds with like the picture of the cell on the right, where that area is white, that like square, as well as the fluorescence versus time graph on the far right. So you basically have the fluorescence, that's a constant rate, and once you apply the bleach, which is really just whatever, a laser beam, the fluorescence in that area goes down. And then there's a recovery phase, which basically is kind of showing you how it returns to that mosaic. So it's like it recovers over time. Because the proteins are constantly like shifting back and forth, back and forth. So that's why in the recovery, you have that bleached area spread out more. And so it's not like a bleached area anymore.
I see.
So these two slides are just there to show you that that over time, a mosaic will form.
Okay.
Which is like a mixing kind of membrane proteins. So that's what FRAP is. And then the next slide, I think Professor tried to maybe put a video of it, but it doesn't work.
Yeah.
And then the run after. Okay, so the rate of recovery is essentially a direct measure of the diffusion rate, which is just the rate at which the membrane proteins diffuse and form that mosaic.
Okay.
So that's the rate of recovery. And then there's extent of recovery, which is just the direct measure of the percent of the labeled molecules that are free to move.
Okay.
So for the results, they have proteins that so essentially for one, they are looking at the rate of recovery, and they find that proteins move more slowly in the plasma membrane than they do in artificial lipids.
Okay.
And then for two, it's essentially like they're looking at the extent of recovery. So essentially 30 to 70% are not free to diffuse. So that's a limit on the extent of the recovery.
I see. Okay. Hmm.
Yep. And the next slide is just talking about proteins. They show different patterns of diffusion. So this is like something called single particle tracking. So it's as if you were to track just one particle. So in the chart, or in the chart, you basically have, if you could imagine just like one particle moving, and you trace the movement and path of that particle, that shows you a certain pattern of diffusion. So for A, you see that it's moving over a very large area. So that's an example of a protein that freely diffuses in the lipid bilayer. Since it's in the lipid bilayer, it can diffuse freely. Whereas B, it's in a much tighter space, and you see it kind of like a square kind of path, a couple of square paths. So that's when it's corralled in a membrane domain by other proteins. So other proteins are kind of inhibiting it from being able to move as freely as it would if it was in a lipid bilayer. And C has the least freedom of movement. It's not really moving that much at all. And so that's an example of a protein that's tethered to the cytoskeleton.
Okay. Let's see. Okay.
All right, then the next slide is talking about several ways that protein movement can be restricted. So we have A, which looks like it's attached to like a membrane domain, but it's on the inside. B is essentially the same thing as A, except it's on the outside. Let's see. C, it appears to be like some type of like junction between two different cells. And D, well, let's see how they describe them. So I'm going to look at the next slide after that, because this one doesn't really describe them. So let's see. It says in specialized cells, cell surfaces have distinct functions and must have order maintained. So we have the apical plasma membrane. That's, essentially the outside, it's the side that is facing like the inside of like I guess like outside is the best way to think of it. So, you have like the villi or whatever.
Yeah.
So that's where regulation of nutrient and water intake happens, regulated secretion, as well as protection. So we have essentially, like the drawing that they have like a glucose sodium symport, it's called, I think. That means like both of them are going in the same direction. And let's see, there's also like a disaccharide that also goes inside. And okay, so this is like the one that is in the previous slide, C. Lateral plasma membrane. So lateral just means like sideways. So the lateral plasma membrane has function of cell contact and adhesion, as well as cell communication. And then the bottom of the cell is the basal membrane, and that's the cell substratum contact. So it's it's contacting. So it's where the cell and the kind of like basement membrane is attached. And then let's see, generation of ion gradient. So it does that. So these are just differentiated functions of the plasma membrane of an epithelial cell. So, there's the apical plasma membrane that has its own functions, lateral plasma membrane, its own functions, and the basal membrane has its own functions.
Okay. All right. So I'm a little confused between 60 and 61. What's exactly happening? Protein movement can be restricted in several ways.
Yeah, so this is just like kind of like drawings of different ways that um, of different types of plasma membranes essentially. Like A is a plasma membrane where there's some type of protein on the inside, B some type of protein on the outside. C is like the lateral plasma membrane, like sideways essentially, like next to each other.
Mm-hmm.
And let's see. And D is kind of like examples of the basal membrane.
Okay.
Yeah. I wouldn't worry too much about that that slide. The one that's figure, well, the one that just has pretty much no description. Yeah, I
Okay.
wouldn't worry about that. The slide after that basically tells you what to what like the what to like, I guess like focus on.
Yeah. Okay. That sounds fine.
Yeah, just essentially like since we up to this point we've only thought of the plasma membrane. Like when we draw a cell, we draw it as like a circle with a plasma membrane on the outside, right?
Yeah.
But now the slide for the epithelial cell shows us that it's not that simple. In an epithelial cell, for instance, there's an apical side, a lateral side, and a basal side. And the cell membrane is different for each of those sides.
I see. Got it. Okay. Next slide then. Why are red blood cells used? A good cell to use to understand plasma membranes. So what does this mean? I know that this would probably be on the test.
Yeah. So apparently a lot of what we know about plasma membrane proteins come from studies with human red blood cells. Why are they good to use to understand plasma membranes? I guess because they don't have, well, whenever you deal with red blood cells, they don't have a nucleus, and I'm trying to connect that to why that would be a good reason to use them to understand plasma membranes.
Okay.
Let's see. An example of plasma membrane structure. Plasma membrane ghosts. Let's see. Peripheral integral. Oh, okay.
All right.
So
Well,
since they don't have any nuclei, it's very. Oh, it doesn't have any other membrane-bound organelles either. So it's very easy to isolate the plasma membrane from red blood cells. So that's the reason why they're a good cell to use to understand plasma membranes.
all right. Explain that one more time.
So they don't have any membrane-bound organelles like. So for instance, let's say we take a regular cell, right?
Mm-hmm.
There's going to be a plasma membrane will be there, which is a lipid bilayer, but there's also going to be a nucleus, which has its own lipid bilayer. There's also going to be, mitochondria, which has its own lipid bilayer as well. So regular cells have lots of lipid, bilayers within the cell. But if you want to study the plasma membrane and just the plasma membrane, a red blood cell allows you to do so because it doesn't have a nucleus. It doesn't have, mitochondria. So it's very easy to isolate the plasma membrane if you have red blood cells.
Got it. Got it. Okay. So how can I put that in a good coherent sentence just so I know?
Well, you could say that.
Like just write it down.
Yeah, you could say that RBCs. Here, I'll put it in the chat. RBCs have no
membrane-bound organelles.
Yeah, have no membrane-bound organelles. So it have no membrane-bound organelles. Only a plasma membrane, which makes it easier to isolate the plasma membrane.
Okay, makes sense. All right, cool. What slide are you on now? Because you usually share the screen.
Oh yeah, I was just looking at
the sample test.
Oh no! I was looking up something about red blood cells, about cytoskeleton. But anyway, let's see. Cytoskeleton and osmosis. Okay, example of plasma membrane structure. Plasma membrane ghosts. Okay, yeah, I was looking at something about icosahedral skeletons. Where did I see that? Oh, triangular mesh of proteins. Let me see. Alright, so typical human cells have a two-dimensional skeleton that supports the outer membrane and a three-dimensional interior skeleton that supports all the organelles inside and serves as a transportation. Okay, red blood cells have only the membrane supports and no internal scaffolding. So they're basically a balloon filled with molecules of oxygen-carrying hemoglobin. Okay. So, here, I'll put this in the chat as well. So, regular cells...
Mm-hmm.
Alright, I wrote, regular cells have a 2D skeleton that supports the outer, aka plasma membrane, and a 3D internal skeleton that supports the organelles inside. Red blood cells, however, only have the 2D skeleton that supports the plasma membrane. Inside is nothing but hemoglobin. Did you test for anemia?
Mm-hmm. Wait, what?
Oh, I mean, you, like, personally.
I believe so. I think they run every test on me at this point.
Yeah.
Yeah, they even did a cancer screening, so. Okay. So, where are you up to now? Which slide? Last one?
65.
Oh, 65. Okay.
So that's just the kind of like geometric
Look
like,
outlook of it on a microscope.
yeah, of the plasma membrane like ghosts. And yeah, and then the components are in that like on that gel. So we have spectrin, which there's a lot of, and that's the name of the meshwork that forms the cell cortex. So in slide 66, you basically have the that like outer layer of the plasma membrane, that two-dimensional outer layer, or rather two-dimensional skeleton that supports the plasma membrane. So that's what you have in the image for slide 66. That's spectrin meshwork.
Yeah.
Yeah. So yeah, you have some actin, you have a junctional complex, and you have spectrin. You also have attachment proteins and transmembrane proteins. So the transmembrane proteins, like, will just let things go from the inside to the outside. So we're looking at that flat image that you see is like the interface, basically.
RBC forms the cell's cortex. Okay. Now last one.
Oh, it is the last one. Yeah. So the plasma that's just a picture of the plasma membrane of a red blood cell. Um,
Mhm.
and so we basically have, let's see, cell cortex, proteins on the inside of the plasma membrane, um, strengthens the plasma membrane. Okay. Um, oh yeah, that makes sense because it kind of like spreads apart in that on that 2D surface. And then I
Mhm.
guess if you have mutations of spectrin, um, it makes the individual anemic, which causes the cells to break in the capillaries. So spectrin is essentially the meshwork that holds the plasma membrane of the red blood cells together. So if someone had a mutation in it, it would cause the plasma membrane of the red blood cell to essentially break apart.
All right. So how can I put that in a sentence that's very?
Let's see. So you could say that since red blood cells, plasma membranes are held together by a spectrin meshwork, if there was a mutation in spectrin, the plasma membrane would fall apart, thereby causing anemia. Since RBCs contain hemoglobin, which, well, I mean, you know what anemia, I guess, is. So, yeah. Since RBC's plasma membranes are held together by a spectrin meshwork, if there was a mutation in spectrin, the plasma membrane would fall apart, thereby causing anemia.
Okay. Cool. Mm.
So that question that says what would happen if if you did frap on red blood cells.
Mhm.
So I'm trying to think of what it would do because when you do frap, you essentially rely on it to form that mosaic again. But I don't know if that is possible because for the red blood cell, everything is held together in that spectrin mesh. So I don't know if it's freely as free to form the mosaic again, because everything is organized and ordered in that like kind of like geometric pattern.
Mm-hmm.
So wait, your test is like on Wednesday, right?
Yeah.
Okay. So let's go over let's open up the that practice test from March 2018.
Yeah. That'll be good.
Was that the last? That wasn't the last PowerPoint, was it?
No, there's 2 more.
There's 12 and 15 as well.
Yeah, I sent those.
Mm-hmm.
That should be there.
Okay, let's open up chapter 12, that PowerPoint.
Okay.
Yeah, I'm looking through it. It seems not that bad. It's just talking about different types of diffusion and stuff.
Okay. Got it. Let's go to another 12. There we go.
Then chapter 15 seems like it goes into, it talks about the Golgi.
Yeah. I'll make up a good amount. Alright, membrane transport.
Yep. Look at it.
Cool.
That's a cuttlefish, right? Yeah. Okay. Ready?
I'm ready. Okay.
Okay. Okay, so second slide, we're looking at the difference between an artificial lipid bilayer and a cell membrane. So the difference is that a cell membrane must... Okay, so lipid bilayer generally is just there to prevent the loss of polar solutes, because if it's polar, it can't cross the lipid bilayer, right?
Yeah.
Okay. Now, if that was completely the case for our cells, then there wouldn't be any way for there to be any movement or exchange of like ions or salts or nutrients or gases like CO2 or oxygen, like the cell would die, right? So a cell membrane essentially is a lipid bilayer, but with certain, transporters and things like that. So that's basically what this is showing you, like between A and B. A is just a lipid bilayer without any transporter, aka without any proteins, because the transporters are proteins. And B is a cell membrane with those protein like the transporters that are made of protein that allow the exchange of, all sorts of things.
Okay. So let me just write that down. All right, cool. All right, I'm ready for the next slide.
All right, the next slide is showing you an example of the synthetic lipid bilayer, essentially just a regular lipid bilayer without any of the transporters and channels and things like that. So it's basically just telling you that membranes are semi-permeable. So what can pass through? Small hydrophobic molecules can pass. So O2, CO2, N2, benzene. And small uncharged polar molecules can also pass. So water, glycerol, ethanol. However, what cannot pass are larger uncharged polar molecules, so amino acids, glucose, nucleotides, and ions of any size. Sodium, bicarbonate, potassium, calcium, chloride, magnesium, those cannot pass.
Okay.
So in our cells, which need those things, there are going to be special things like transporters and membrane channels that allow the larger uncharged polar molecules and the ions to enter and exit the cell.
Okay. This is in a regular cell, yeah?
Nope. That picture is in a synthetic lipid bilayer.
Okay. And what's the purpose of this exactly? I'm not sure. I'm not understanding.
So I wrote in the chat, lipid bilayer enables the transport of small hydrophobic molecules and small uncharged polar molecules. However, it doesn't allow the transport of the larger uncharged polar molecules or ions. These require membrane transporters or channels, which the cell has. So the cell has that, and an artificial lipid bilayer does not. So that picture is just for, like, don't even, like, worry about me saying artificial or it saying synthetic. Just think of it as a lipid bilayer. So, you know, our cells have lipid bilayers, but it's not the only thing our cells have. If it was, then, you know, it would be like, you know, this slide, which basically shows us that ions can't get into the cell then. Or, you know, amino acids and stuff.
Okay.
Yep.
Okay, continue.
Mm-hmm.
Oh, my mom says hi, by the way.
What's up?
My mom says hi.
Oh, yeah. Tell her I said hi as well.
Mm-hmm. Which regular cell has?
Training stopped.
Okay. I see. Okay, so the artificial bilayer is just the example that they're using.
Yeah.
but a regular cell allows those things to move through, but they need specific channels.
Yeah.
So yeah, I get it now, because the larger, because what they're basically trying to say is that the small hydrophobic molecules and uncharged polars go through without an issue, without anything special
Yeah.
and stuff, and then the larger stuff needs some special things in order to get through. So it's just showing the simplicity of each. I get it. Okay.
Yeah, larger, or if it's an ion.
Yeah.
Okay. So the next slide is basically showing us, and you might remember this from when we did neurons with action potentials and stuff.
Yeah.
So there's going to be, so essentially we have an extracellular, so there's a sodium concentration outside of the cell that's really high, like 145, like millimolar. And then there's like a potassium concentration on the inside, which is basically the same. So essentially there's approximately equal quantities of positive and negative charges inside of the cell and outside of the cell. So, and then tiny excesses of positive or negative charges do occur and have important electrical effects. So that's like an action potential or something. So, don't really need to. Yeah, don't really.
Okay.
Yeah, need much else from that.
Each cell membrane has its own characteristics.
Yeah. So.
Carrier proteins. Yeah. I get this.
Yep.
So the pyruvate right there, stuff that has to do with glycolysis.
Yeah. So you see how. So this is kind of like, you know, so a regular cell that isn't a red blood cell, right? So just to go from like the previous PowerPoint, like if we wanted to study the plasma membrane and we used a regular cell like this, we're gonna have like the membrane of the mitochondria, the membrane of the lysosome, you know, so many other things. But the red blood cell only has the outer, you know, plasma membrane. So that's why red blood cells are ideal if you want to study just the plasma membrane. But yeah, this is a regular cell, so you have, you know, a nucleotide transporter, a sugar transporter, amino acid transporter, sodium potassium pump. Yeah. You know, H plus pump, pyruvate, etc., right? So, yep.
Yeah. The NA pump has to do with like action potentials and stuff, yeah?
Uh-huh. Yeah. I think they yeah. So, if something goes from a high concentration to a low concentration, it's diffusion. If it goes from a high concentration to a low concentration using a transporter or a channel, it's facilitated.
Oh, so it's facilitated diffusion.
Yeah. It's just the same thing as diffusion. It just requires a transporter or a channel to facilitate that diffusion process.
Yeah, osmosis is the diffusion of water.
Yep.
Awful. Okay.
So.
All right. Can you say that? Can you say that part just one more time?
Oh yeah. So just basically, diffusion doesn't require like anything. It just can go freely through the membrane or whatever. But if it has to go through a transporter or a channel, it's called facilitated diffusion.
right? Specific channel or something. It's facilitated diffusion.
Yeah, channel or
A
transporter.
specific channel on transport.
Yeah, and then we're going to differentiate channels and transporters.
Okay.
So, a channel can either open or close, and it can go. Stuff can go inside, stuff can go outside, and you kind of see that in that drawing over there. But transporters are not like that. It's more like a
They
solid.
work like
Yep.
what do you call it? Enzymes almost.
Yep.
They have to bind to the site, and then
Yep.
that site activates and stuff. I remember this is the stuff that you taught me when we were doing our first semester together.
Mm-hmm.
You were basically telling me to think of transporters as enzyme sites.
Mm-hmm. Yeah.
Conformational changes, which is just basically binding sites.
Yeah. Conformational change is a change in shape.
It is usually done via a binding site that must be activated. To facilitate the diffusion. Facilitate the diffusion is a good way to put it.
Well, both channels and transporters are examples of facilitated diffusion, but like.
Oh, okay. That must be So I said this is usually done via a binding site that must be activated in order for transports to happen.
Yeah, think of like a channel is like a faucet, which you just can turn on and water just comes out.
Yeah.
But think of a transporter as like a vending machine.
Yeah.
It's not like a free kind of flow. It's like something like a solute has to bind to the transporter, and the transporter changes shape, and it will do something like let the solute out or something. So it's slower,
Yeah.
Kind of like enzymes. Okay. Influx.
Influx and efflux. I remember
Yep.
this.
And active transport is when it requires energy. So, if something's going from a low concentration to a high concentration, that's going to be an example of something that requires energy. So that's active transport. Yeah, they talk more about this in some later slides, so we can just keep moving along.
Okay.
Because I see like a slide later on that summarizes those in like just one.
Yeah. Fine.
So okay, let's look at slide 9. So this is an example of an electrochemical gradient.
Okay.
So, electrochemical means that there's an electrical aspect, which you kind of see with like there being a positively charged outside area and a negatively charged inside area. And the chemical gradient part, which refers to the ions on the outside and on the inside. So the electrochemical gradient. So for A, electrochemical gradient, when voltage and concentration gradients work in the same direction. Yeah.
Yeah, there's
So like
seems to be. All right. Well, that's
yeah. So basically, like the voltage gradient is positive on the outside, negative on the inside, and the concentration gradient of the chemicals, aka the ions, is positive on the outside, and like negative on the inside. So it's going, from the outside to the inside. And then for B, the one on the right, it's just going in the opposite direction.
okay. Well, why is the width smaller on the one that's on B? When they're working in the opposite direction, but they yield almost the same on each side.
Just because, if it's going to follow diffusion, right?
Yeah.
So for A, there's a higher concentration of positive ions on the outside than on the inside, right?
Okay.
So it's going to go from a high concentration to a low concentration for diffusion. So that's why that width is greater, because there's a higher concentration of positive ion on the outside than the inside, right? So it's going from high to low.
Yeah.
And then and then on the right, it's going high to low too. But I guess this is just like because let's see. Oh, I get it. So on the left, if you move positive ions from the outside to the inside, it's going to make the inside more positive. And, kind of making it closer to equal. Whereas if you take positive ions from the inside to the outside, you're just making that gradient even greater. You're making the positive part on the outside more positive, and the inside part more negative.
So in A, since it's both on the same side,
Yeah.
and stuff, the gradient is going to be larger because the voltage would be greater.
So in A, so we have positive on the outside, negative on the inside, and if we have, and it's going to, every time there's a gradient, like, it's going to want to return to equilibrium. So if the outside is more positive and the inside is negative, it's going to work in such a way as to make the outside and the inside almost equal.
Okay.
So if positive ions from the outside, which is more positively charged, go from the outside to the inside, the outside will become less positive and the inside will become more positive. So it's going to return, it's going to come closer to equilibrium.
Yeah. Okay.
But if you're moving positive ions from the inside to the outside, then the inside gets more negative because it's losing the positive ions, and the outside, where the positive ions are going, gets more positive.
I see.
So it's almost like a battery. Like a battery works because there's a concentration and voltage gradient in a battery where one side is more positive and the other side is more negative. And if you connect a wire from the positive side to the negative side, you're going to have electrons move to make the positive side more negative and the negative side more positive until they're equal and neutral. That's when the battery dies.
Okay.
But we don't have to. It's not really that important.
Okay.
Like, it could just be arbitrary in this case.
I get it. All right.
But generally speaking, like, you want the outside and so if you remember, like, the slide 4. Where it says approximately equal quantities of positive and negative charges inside and out of the cell.
Yeah.
So that's what you want, right? That's like the equilibrium.
Yeah.
So if you have the outside that's really, really positive and the inside that's really, really negative, you don't really want that. You want the outside and the inside to be kind of the same. So how do you achieve that? By bringing positive ions from the outside inside.
Okay. Electrochemical gradient.
Yeah, so a gradient just means that there's a differential. There's a yeah.
Okay.
And things are going to diffuse in such a way as to eliminate that differential and to make it equal.
Okay. So I guess what I'm just trying to grasp at here, because it's just the width of that green arrow, I'm like really, I think it's just throwing me off.
Here, check this out. You can see my screen?
Uh-huh.
Alright, I'm going to draw something. So remember when okay. For like, let's say this is negative 70. Whoops. So this is a, let's say this is positive 40. So this is like a depolarization thing for a neuron. So a neuron is normally at negative 70 millivolts. All right.
Okay.
So that basically means that trying to think of like how to just make this turn into a wait, I could just do this. Wait. Okay, let me go back. Alright, so inside of a neuron is negative 70. Outside is, I don't know, like positive whatever, 70, let's say, okay? So that's its resting state at negative 70. And when an action potential happens, it causes the opening of sodium channels. So sodium on the outside is like 140 millimolars, right? And on the inside, sodium is like 5 to 15.
Mm-hmm.
Let's just say 15 millimolars, right?
Yeah.
So if a sodium channel opens up, which direction is the sodium? So this is sodium plus, right? Ion. Which direction is it going to travel?
If it's what?
If the sodium channel or pump opens up, which direction is sodium going to travel?
It's going to travel going to be down?
So wait, this is the...
I mean, up, sorry.
Yeah. This is the inside of the cell, and this is the outside. So on the outside, there's 140 millimolar concentration of sodium. On the inside, 15. So if you opened up that channel, which direction will the sodium ions go?
You can go down.
When you say down, you mean what? There's only outside of the cell and inside of the cell, so...
Oh, go outside of the cell.
Well...
Oh, wait, no, but if you're trying to charge it up, you go inside, yeah?
So if you open it up, diffusion, which is high concentration to low concentration, right?
Yeah, it's higher concentration outside of the cell, so... what do you call it? It would go from out to in.
Yeah, it'll go inside, right?
Yeah.
So you're basically having then positive ions going from the... So outside is already positive, right?
Yeah.
So it's like this, right? Where the outside is already positive, right? And the inside is already negative, right?
Yes.
And once you open up this pump, there's a lot more sodium positive ion on the outside than there are on the inside. So a bunch is going to go inside. So that's why in an action potential, when you have depolarization, it goes from negative 70 all the way to like positive 40. Here. This right here. Oh, cool. It like lines up almost. But this is the action potential. So at negative 70, that's where it is. And something happens, some stimulus, which causes the sodium channel to open up, and a bunch of sodium ions get flushed inside. So much so that it causes the charge of the inside to go from negative 70 all the way to positive 40. Right? So that's what this is an example of. Right?
Yeah.
So that's why, it's going from the outside to the inside, rather than, like in this example, where positive ions within a negative space go out. That would be essentially the same as saying that sodium ions should go from this inside region of low concentration outside to the high concentration, which isn't the case, right? So, yeah, so, like, common sense would dictate that this doesn't happen at all, but it happens to a small extent. And the reason for that is the reason that this stays at negative 70, because I know I said before that it should want to go to equilibrium where it's 0 volts on the inside and 0 volts on the outside, but it maintains that concentration gradient using something called leak channels. So this still happens even though this goes against diffusion. It still does that through leak channels. But yeah, that should explain why this is going to happen.
Got it.
Yeah, it's like this is like sodium concentration on the outside. Whoops. And then, it's a lot more than it is on the inside.
Okay. Let's move on.
Okay. Alright, and this is the slide that I said, summarized a bunch of the things. So, there's simple diffusion, which is just through the lipid bilayer. There's facilitated diffusion, which is through channels or transporters. And then there's active transport, which means that it goes from low concentration to a high concentration. That is the one that requires energy, and that is done through pumps.
Okay.
All right. Yeah, and smaller is faster, etc. Okay, so I guess then we can
Oh, okay, it's gonna go through the 4 thing. Okay.
yeah. So ions go through the channels. And these are the types of channels. There's gated channel, which open and close with the proper stimulus. And bidirectional.
Wait.
Yep.
No, we skipped over a couple.
Oh yeah, I just skipped over diffusion through membranes. That just basically high concentration to low concentration. And it just goes through the lipid bilayer.
Okay. And what's the partition?
It's just it's a measure of polarity, which is just a ratio of solubility in a nonpolar solvent to its solubility in water. So basically, let's say partition coefficient is proportional to, or rather it's inversely proportional to polarity, because it's a ratio of how soluble it is in a nonpolar solvent compared to a polar solvent like water.
Okay. Okay. And then it's just high concentration and low concentration, yeah?
Yep. That goes the same. That's the same for everything that is passive over here. So
Okay.
basically all of that.
Oh, everything in these four?
These
Out of the four
three
things?
over here. Basically, if it's from
Oh. Okay.
yeah, if it's from
Okay.
high to low, it's passive, because it doesn't. It just goes like that automatically. It's like if you're in a packed subway car, and is it harder to get out of a packed subway car or to get into a packed subway car?
It's harder to get in.
Exactly. So that means if you're going from a low concentration to a high concentration, it takes energy. Whereas, it doesn't take energy to get out of a packed subway car because people are, pushing up against you and stuff. It's harder to stay in and get in. So that's why if you're going from a high concentration of anything to a low concentration, it's called passive. So if it's going from low to high, then it's active.
Okay. If it's high to low, it's active?
No.
If it's high to low, it's passive, and then if it's low to high, it's active?
Yeah.
Okay.
Like, if the subway car is packed, the concentration inside of the subway car is high, and outside is low. So it's easy to have all those people push up against you to push you out. It doesn't take any energy for you to get pushed out. It takes energy from you to, get yourself into that high concentration subway car.
So basically A, B, and C are from high to low, and then D is low to high.
Yeah, correct. And that's why you
Okay.
see as D goes from low to high, it takes energy. ATP is used.
Yeah. I see. Okay.
Yeah.
All right, so next one.
Yeah. So ions pass into the cell or inside and out of the cell through ion channels. And there's gated ones and bidirectional ones. So a gated ion channel can open or close. So in that example for a neuron, there's a voltage gated sodium channel, so that can open, letting sodium into the cell. But there's also bidirectional ones, which means that it can go into or out of the cell. It can go in either direction.
Yeah. Well, that was a pretty simple one. Okay. That's pretty self-explanatory. It's just that, right? Or is
Yeah,
there more?
we're going to talk about different categories of ion channels after.
Okay. Go to it. Oh, okay, there's different ion channels.
Yeah, and you know, before that, let's just talk a bit about specificity. So you see how hydration of a sodium ion is different from that of a chloride ion in terms of how the water molecules arrange themselves outside.
Yeah.
So that is how an ion channel can have a selectivity filter. So, for instance, this one is a potassium channel, which only allows potassium ions to go through. And, you know, it can do that based on looking at the arrangement of the solvation of the ion. Anyway, we can move on to slide 18, which talks about the different categories of the ion channels.
Okay, but wait.
Yep.
This slide 14, the fusion of ions through membrane channels. What can I copy from that? Just the gated and the bidirectional stuff?
Yeah.
Okay. Ready. And hydration of the chloride ion and the sodium ion. Were you trying to explain me from that again?
Oh, just that. So the gated channels can select for a particular ion, and it does that by so in this diagram, right, we have the potassium ion here and water molecules around it.
Mm-hmm.
So, you know, it goes into, you know, one of the pores or whatever, and here in this region, essentially there's no more water around it.
Mm-hmm.
And that is where that's where it basically can tell the difference between like whatever type of ion it is. Like it can tell the difference between like a K plus ion or not, or like like a sodium ion. So all you need to know is just that, let's see. Here, so I'm going to write some notes here so that can help you with your notes. Ion channels, so gated and bidirectional, on or off. Oops. Okay. So this should help you with knowing like what to write and stuff for notes. So ion channels, gated and bidirectional. Gated can be on or off depending on the ion. Maybe I should write depending on the identity of the ion, and then through specificity filters.
Okay, I put open and close the popper stimulus, so I can just reword it as that.
Yeah, because this way we also have the selectivity filter stuff. So like, you know, I kind of like forgot that like note taking skills was also something that we worked on because we haven't, like, I kind of forgot about that. So this is just me kind of taking that slide and then the slides after and putting it all into just one thing for you.
Okay, so I put can be on or off, or quote unquote open and close, depending on the identity of the ion and
Yeah.
specificity filters.
Yeah. Like the double arrow thing essentially means like through that mechanism.
Okay, got it. No. Okay. And then this hydration of the ions and stuff just has to do with the positive and negatives.
That we don't have to go into that too in depth at this time. All we need to know is at this point is just that the specific channel can tell what ion goes through it or tries to go through it.
Yeah, alright, so the potassium channel over here is basically trying to say this is like how a gated channel works.
Yeah. Oh crap. Sorry, it's selectivity filter.
Oh, okay, not specificity filter. That's fine. Almost means the same thing.
Yeah.
And then I'm just going to put on this example of a gradient channel. Okay, cool. All right, next. Potassium channels are unsolvated, which means the water's removed.
Yeah, go to slide 18.
Yeah. Okay. Okay, major categories of gated ion channels. Okay, got it. Okay. And voltage gated. Okay. Good. And this goes through something called patch clamping.
Yeah, we don't need to really focus too much on that, but that's just that's just an example of them. Essentially, what they're doing is just using it to monitor the activity of the ion channel. So basically, what they're doing is they attach they attach this like electrode essentially to a part of the membrane where an ion channel is, and they see how, like the they run a current through it, and they just basically use that to monitor it. But basically, all you need to know is that, so you see how I wrote it in here.
Mm-hmm.
Voltage gated opens due to change in voltage.
Mm-hmm.
Ligand gated opens due to ligand binding. So, example of that are neurotransmitters. And then there's stress, which is just another word for mechanically gated, and that opens due to mechanical stimulation. So example of that are hair cells, which are in our ears, and that's how we can hear.
Hair cells in the ear. Hmm? No. Yeah, neurotransmitters. Okay. I see. Oh, okay. Aren't headaches caused by something wrong with ion channels and stuff?
There could
Or
be like
migraines?
because
Because this looks exactly like an example that they would use to show like the impulses or electrical impulses that are in your brain or stuff that cause migraines.
well, I mean, generally speaking, like a headache will be, well, like what exact, like are you talking about like an EEG or something?
Yeah, I think so.
Yeah, like, there's always going to be, you know.
Yeah, like ion receptors and stuff like that.
Mm-hmm.
Yeah, and then there's like malfunctions in those which can be more prone to headaches or migraines.
Yeah, I'm thinking of with migraines. Yeah, there's like a rapid. There's going to be a rapid wave of depolarization through a certain part of your brain. I mean, if you have like visual auras, then it'll be through like your occipital lobe, which is like back,
Yeah.
which is
The back
responsible
of the
for
brain.
responsible for vision.
Yeah, it's like on the back right.
Yeah, that's what like Neanderthals have much. They have much larger brains, but it's predominantly their occipital lobe because they evolved in low sunlight conditions, so it helps them see at night.
Yeah. Mechanically gated. That's understood.
Yeah, so an example of that are the hair cells, which are in our ears. So, like, when they can detect, so when there's sound vibrations of different frequencies, it'll vibrate, cause the hair cells to vibrate, which causes them to mechanically move, and that causes these mechanically gated channels to open up and send, nerve signals to the part of our brain that interprets the sound. Right.
Stress gated ion channels lost here. Got it. Okay. All right. Next is difference facilitated transporters.
Oh, okay. Yeah. So basically. All right. Let's see. I mean, like, this is kind of like stuff that you kind of know. I mean, facilitated is just, it goes through a transporter, right? So This is basically just, instead of channel, like we went through facilitated diffusion with channels, and now this is just with transporters. So the passive thing, like, that goes without saying. Now let's see what to take away from this.
Well, there's passive facilitation, which doesn't require energy.
Basically, everything is passive unless it's a pump, like an active, active transport. But yeah, looking at the glucose transporter is a good example. So that's on slide 30.
Bidirectional depends on the concentration. And the concentration. And you said most facilitative transport is passive.
Yeah, all. So that definition makes it so a lot of this stuff is redundant, because if it's passive, right, it's going to have to be from a high concentration to a low concentration. So bidirectional depends on concentration, that goes without saying, right?
Yeah.
Because if there's a higher concentration on the inside, it's going to be going out. Higher concentration on the outside, it's going to be going in.
Yeah.
But the main thing to take away, I'll put it here. I'll put it in the notes.
So you said facilitative transport is always passive unless it is through a pump.
Yeah. Like,
such as a glucose pump.
Well,
or such as a sodium pump. Any.
Well, we'll come to that when we come to that.
Okay.
Just yeah. So about transporters, basically what happens is okay, so the thing, I mean, I was going to call it a ligand, but alright, I guess they call it solute. So solute basically binds to transporter changing its shape causing it to transport the solute or another solute into or out of the cell.
Okay. Okay, so the transporter solute binds to the transporter, changing its shape, causing it to transport the solute into and out of the cell.
Yeah, and then in parentheses I wrote, or another solute.
Binds. Alright, got it.
Mm-hmm.
Glucose transport changes same around in the medium without the glucose. Glucose binding. This is the shape change. A side of the membrane with highest glucose concentration will tend to bind most of the GLC.
Oh, I added something for transporters. Can be
Mhm.
regulated. AKA, by feedback.
Can be regulated by feedback.
Yeah.
Like, okay. Alright, I'm back.
And then I drew some pictures of some stuff that might as well, because they're gonna go over it at some point. But these are just kind of different types of transporters. There's a uniport, which just has this solute going in or out, right? Symport, which means that
I remember this.
yeah, two of them go in the same direction, and an antiport where they go in opposite directions.
Forms of transport. Three different forms of transport direction. And that's uniport, which is one way. Symport, which one way but with two. But with more than one, right?
Yeah.
And then antiport is bidirectional, right?
Correct.
Is that a good work for it?
Yep. Well, actually, no, because channel we described as bidirectional.
Oh, okay, so antiport is basically...
Antiport just means that one goes in, like, one, whatever.
Yeah, one goes in, one goes out.
Yeah.
Got it. Okay.
All right. Now I've already started with active transport. Essentially, I wrote low to high, which requires energy.
Mm-hmm.
Examples are pumps and leak channels. So the leak channel is kind of like what I was talking about before, where in the neuron, a negative 70 millivolt voltage exists inside of the cell, and it doesn't want that to happen.
And then I put the glucose transporter example, which is that's the slide. Pretty self-explanatory.
Yep.
And it's the same thing with insulin. Okay, so that's cool then. Yeah, same thing, regulation, endocytosis. Yeah, okay. This is a lot of the Bio 1 stuff that we were doing.
Mm-hmm.
Alright, what are you putting for here? Active transport, okay, so that's low to high. Ion pumps and leak channels?
Oh, yeah, sorry, leak channels.
Yeah.
The leak channels maintain concentration gradients.
So those are like the forms of active transport, yeah?
Mm-hmm.
Okay,
Yeah,
so
like
ion
examples
pump,
of, transporter, yeah.
ion pumps, and then leak channels.
Yeah.
L-E-A-K, right? Not like leak the vegetable.
No.
Okay.
Yeah, so the sodium potassium pump is an example.
Sodium potassium pump, ATP ACE. That's understood, yeah.
Yeah, so the sodium potassium pump it basically, so we know that there's more sodium outside than inside, and we know that there's more potassium inside than outside. So sodium is going to want to come inside of the cell, and potassium is going to want to leave the cell. And the sodium-potassium pump, or whatever, ATPase, has this uneven, like, kind of distribution, where for every 2 sodiums that it pumps out of the cell, for every 3 sodiums that it pumps out of the cell, 2 potassiums get pumped into the cell. And that unequal amount means that there's going to be a greater, it's going to be more positive outside than on the inside. So that kind of keeps that concentration gradient. It keeps the inside negative.
Okay. I just understood the sodium potassium the electrochemical gradient. Okay. Yeah, it changes shape. Okay, then that makes sense. Okay. The pump is unidirectional. Yeah, that would make sense.
Alright. Okay, I guess you can call it electrogenic. Because it gen like means beginning, causing whatever. So it causes the, electrochemical gradient in this case.
Affinity. What does that word mean again?
How much it likes something.
How much it likes something?
Yeah. Where do you see that?
Okay. Phosphorylation induced shape.
Oh, yeah. So it just changes the
shape happens because of phosphorylation. Yeah, okay.
Yeah. So it alters the affinity, how much how much it likes something, or how likely it is to bind to something. So basically, that's more of a mechanistic thing that, we don't have to focus too much on this first pass. But essentially, you might be asking yourself, how is it unidirectional? I mean, if sodium's going inside and potassium going outside, seems like it's bidirectional. Well, the reason that it's unidirectional is because when it's phosphorylated, so it naturally has a higher affinity for sodium, and when it gets phosphorylated, it has a higher affinity for potassium than it does for sodium. So that's what is meant by that. So it lets go of the sodium, which leaves the cell, as it changes its affinity to be more towards potassium. That's just the technical kind of stuff. But for this first pass, we just need to know essentially just what these transporters and stuff do essentially. I see in slide 43 it goes over the uniport stuff, which we've already, we did
uniport, antiport, and all that stuff.
Yeah. So I should probably.
Oh, it's
put
carrier
these-
method. It's carrier mediated.
What's up? So I should
It
put these
says-
under the category of coupled transport. These are coupled transport, which means like one thing is coupled to another thing. So see how I wrote that over here?
Yeah. Oh, so I should put this then in the, because I have this for number 3 with the facilitated transporters. So I just switch uniport, symport, and intport all to the active transport.
No, because that's still
Oh, that still holds true.
like the thing is that transporters will be a facilitated transporter. Like, well, it'll be facilitated diffusion if it's high to low, but it'll be a pump if it's low to high.
Got it.
But the coupled aspect of it is what it's kind of like using like the momentum or the energy of something. So for instance, let's say we're dealing with a pump, right? It requires energy, so it's coupled to ATP.
Yeah. Okay, I got it. It's,
Yeah.
Okay. Yeah, it would be coupled to ATP because it needs the energy in order to
Mm-hmm.
be
Yeah.
used.
So that's
Yeah,
like
anything that pretty much requires energy would require ATP.
yeah. So that's the case. However, let's say that we couple it to something that goes down its concentration gradient. Like, for instance, let's say that there's a lot more sodium outside than inside, right? So
Yeah.
sodium should go naturally from outside to in without requiring any energy. But we can couple that to something that does require energy. So we can take the excess, from sodium going from high to low, and we can use that. We can couple that to something that requires energy. So it doesn't have to be ATP all the time. Let me see. So here, let me see if I can give you an example. So, let's say we have. So, okay. Well, that's how the ATPase works. So, basically, sodium goes down its concentration gradient, and it uses that to make alright, let me just look through the slides. Let's see.
Alright, so can I try and answer this question with you? So it says if 3 Na plus ions come out for every 2 K plus ions in, what does this do to net charge on each side of the membrane? So what do you call it? Wouldn't the inside become more negative?
Yeah.
And then the outside become more positive?
Mm-hmm.
Okay.
Yeah.
Oh, it says NX. Never mind. I had the answer there. But okay, I'm glad I was able to put that.
So, okay, so think of the sodium outside of the cell like water behind a dam. And you can use a dam to generate energy, like electricity and stuff, right?
Yeah.
So that's kind of what they're talking about when it comes to coupled things.
Yeah. I get it.
Okay.
I understand the coupled transporters thing now. Because what they're talking about here is like it can function via like with for uniport and antiport, for symport and antiport, it can function as two things.
Mm-hmm.
Like it can what do you call it? Exchange two different elements or things that's needed.
Yeah. So let's say for a symport, right? Let's say that this red one over here
Yeah.
is sodium. And
I get it. Okay. Yeah, the couple transporter, so it only applies to symport and antiport. Because uniport would be, for example, quote unquote, a glucose transporter, because it's not a pump. Okay. I think I'm grasping it pretty well.
here, so here, check this out. So let's say that so on the outside of the cell, sodium concentration is 145. Inside of the cell, sodium concentration is 5. Now, here, check out what I drew over here. So, let's say we have some other compound or ion or whatever called X. On the outside, it's 0, and on the inside, it's 500, okay? So, diffusion would state, or facilitated diffusion, would state that sodium would go from which direction?
Out to in.
Okay, so sodium should go like that, right?
Yeah.
What direction should X go?
It should go in.
Well, how much is it on the outside?
Oh, that's true. So no, it would stay out.
Well, so there's 0 of it outside and 500 inside. So diffusion is what concentration to what concentration?
High to a low concentration.
So it
in
should
this
go.
case,
So it should go in which direction?
down.
Wait.
Or it should come in.
Well, that's also down.
Okay, it's high to low. Oh, because you have two X's written down there. Is that a Y or an X?
Oh, my bad. Well, this is X and this is X. So X on the outside is 0. X on the inside is 500.
Yeah. Okay. So then X should be coming out.
All right. That should be coming out, right?
Bottom X should be coming out.
All right.
Let's say X2 should be coming out.
So it should be coming out, right? So in order for it to
Yeah.
go in, that's going to require energy, right?
Energy, yeah.
But let's say that this happens to be a symport. So now it's going to take the natural diffusion, facilitated diffusion of sodium, and it's going to couple that to this one to bring it in. So here, check out what I drew. So that's if this is a symport, okay?
Mm-hmm.
So X should not be moving in this direction, right?
Yeah.
But for a symport, it's coupled to this sodium transport.
Hmm? Okay, so oh, it's couple. I understand now. Okay.
Because normally speaking, X
Yeah.
should not
Wait,
be.
can I
Yeah?
can I see if I can explain it to see if I have it right?
Yeah.
I guess. Okay. So basically, what you're saying is that the Na plus on the left, right, and X on the right. Well, since X basically has zero energy attached to it, but Na has all this energy attached to it, it can couple itself so the Na uses its energy to come in,
Exactly.
and then what do you call it? It allows the 5 and the 500 to come out as they need.
Yeah. Because X, for it to go from 0 to 500, requires a tremendous amount of energy. And
Yeah, so it would need to be coupled with something
this
else
thing
in
that
order for
doesn't
it to
require
transfer
energy.
over.
Yeah.
Yeah.
So that's what it means by coupled transport by gradient-driven pumps.
So basically, it
Yeah.
like transfers over.
Yeah.
Power.
So this is just a made-up, example, but this is basically the sodium is the thing that goes down its gradient, and the X thing, goes in the opposite direction naturally. So it's going to need energy for it to go in this direction. So it can be coupled to that. Mm-hmm.
That's why this slide number 46 over here, glucose Na plus import protein and stuff. That's like basically how it works, since the glucose would need the energy from the Na plus.
Yeah.
Because it's not an ion.
Yeah.
I get it. Okay. That works then.
Yeah, because the glucose gradient, on slide 46, there's more glucose inside than outside. So it should go out, but it goes in because it couples it to the sodium gradient.
Yeah.
So yeah, they described it by saying glucose is dragged into the cell along with the sodium.
Basically, coupled transport is a transfer of energy.
Yeah, I think of it as like using the momentum of something that's moving naturally to kind of like slingshot.
Using the momentum of something else and latching onto it so you can slingshot with it.
Yeah, I don't know, or maybe like
yeah, I mean that's an okay example. That's good wording, because then I can put it all together. I just need like words. I don't need explanations.
Mm-hmm.
Because if I know the explanation, I just need a
Yeah.
what do you call it? A trigger word.
Yeah. Look what the cat dragged in. No, you know like the phrase.
Yeah.
Yeah.
Co-transport. Okay, so we basically went through this.
Yep.
Okay. And it was a much easier explanation. Membrane potentials and nerve impulses. We've basically gone through this. When ions of one type cross membrane, the charge difference creates a membrane potential. Yeah.
Yeah, the leak channels, okay. Alright. nerve cell stuff.
So you basically think of membrane potential as almost like, you know, like how the Flash, whenever he runs around something or trying to like create energy and stuff like that for the guys to power stuff up and whatever, they use his momentum and the energy he's passed on from the movements he's made and stuff, and they use that as the potential to start up something else.
Yep. Because he's going to be carrying an electric charge. From the friction of the air.
The movement and the friction of the air.
Mm-hmm.
Yeah. So he creates a membrane potential by the movement.
Mm-hmm. That's why. Because pretty much like the momentum thing will always exist, but like if he was in space or something, like the electricity part wouldn't exist. Because like when you think about it, lightning comes from the friction of the water vapor and the air particles.
Yeah.
So that's how he's able to like generate electricity essentially, and shoot lightning bolts and stuff
with the air. All right, so I can just think of memory potential like that.
You mean like coupled stuff or?
Yeah, couple things, or the leak channels.
So the leak channels are more like just like so remember how I said that, so see how there's 145 outside, 5 on the inside? Naturally, it's going to want to equilibrate. It's going to want to have an equilibrium of the same concentration of sodium inside and outside.
Yeah.
So that's how it should work, but it doesn't work like that because of leak channels. So like that means that some sodium is just always leaking out of the cell, or potassium. That's the thing that maintains a concentration gradient. Like imagine if you just had like the static electricity on you like 24/7 or something like that.
Yeah.
But it's like a battery kind of. Well, exactly like a battery.
Okay. Resting potential. Let me get some of this. Okay. Skipping over because it's not really necessary per se. Okay, and the X and hillock.
Like, so honestly, like thinking of it as a battery is the best way. So like a battery, right, has positive over here, negative over here, right? And when you put it in something, it connects it to the circuit, right? And over time, right, electrons basically, right, are going to be moving. Electrons are going to be moving from the negative part to the positive part, right?
Yeah.
So after enough electrons, the positive will become like neutral, and after the negative side loses enough electrons, it's going to be neutral, right? So when the battery is neutral, it's dead.
Yeah.
So in order for a battery to work, it has to have that electrochemical gradient.
Yeah.
And just like the thing with the cells, we exploit that electrochemical gradient. We couple it by putting the battery into things. We couple that gradient to generate electricity. Okay. Squid. Oh, I thought that was a cuttlefish. Alright, whatever. Squid giant axon. Axon is big, not the squid. So it is not the giant squid axon, and you needn't be afraid. Alright, action potential stuff. What?
No, the joke.
Oh, yeah. I don't even think they're that big. Colossal squid size.
No, they aren't that big. Well, regular squids aren't that big, but I don't know about a colossal squid. That might be
It's
totally...
not that big right here. Anyway. Propagation, okay.
Okay. So... Alright, since we're at action potentials over here and stuff, and there will be the last 15 slides here, and then we have the rest of the slides tomorrow.
Yeah.
And stuff. Do you want to just, what do you call it? Start with this tomorrow?
Yeah, let me just see this for a sec.
It might take a tiny bit of explaining, and then we'll have the other one. We only have 20 minutes left. So if anything, we just transfer this 20 minutes to tomorrow. To do 12 to 3. Like, no, 20 or something, or whatever.
Hold on, let me see something.
11, 4, 320, something like, I don't know. Oh, man. Are you seeing how much they asked about action potential?
Oh no, I wanted to ask you some questions over here. What's the orientation of cellular membrane associated carbohydrates?
Oh, can you expand the page?
Which page?
On your screen.
This?
Yeah, because I don't have it uploaded here, and I might as well just look on with you. Yeah, do it a little more. Got it, okay. What's the orientation of cellular membrane? No. It's B.
It's what?
I think it's B.
Yeah, good. What about 3? This one over here.
How can passive ion transfer carriers be distinguished from active transporters? Gradients, both gradients, such character carriers of a class. No, it's not B. Passive carriers don't consume ATP energy. You know, beta sheets, nutrients, membrane, no. Active carriers contain alpha helices, no. Passive carriers transport sugars, active carriers transport ions. No, because there still happens with sugars. Because ion sugars and amino acids need transport. So I want to say A.
Good. So I was a little worried about if you understood the concept of a gradient.
Yeah.
But a gradient, in your words, tell me what a gradient is.
Huh?
In your words, tell me what a gradient is.
Basically, how where the what do you call it? The thing that's trying to move along or whatever passes through.
So a gradient is simply
Uses the
a
passes through.
differential, a difference in. It could be for a concentration gradient, it means that there's a difference.
Difference in concentration.
So that means like there's high on one side, low on the other, right?
Or a differentiation of charges.
Yeah. So diffusion,
yeah.
right? If there's high on one side and low on the other, and diffusion is high to low, at the end that gradient is going to disappear, right?
Yeah.
That's what I was trying to show with the battery thing. So
yeah, it's only going to be there for one, but the active transporters can build gradients because they have energy.
Well, so just like it takes no energy for you to exit a subway, right?
Yeah, it takes energy to get into one. So
Yeah.
you need to build up your strength to get in.
Yeah. So, okay. So if there is a gradient, right? So why is it that inside of a neuron, there is a -70 charge on the inside? Why isn't it 0?
Say that one more time.
In a neuron, like a nerve cell, why is there a -70 charge on the inside? Why isn't it 0?
Well, because nerves move via an action potential and stuff like that, and there's going to be a -70 charge because there's still, what do you call it, energy left from the movement.
Well, the nerve cell will be at -70 before any type of action potential happens, right?
Yeah.
The action potential, if anything, makes it more, equal to the outside, right? Because the outside is positive.
Yeah.
So, just, the simple thing that I want you to get from this is if there's a lot more... Wait. Okay, it's this, right? So, -70, +70, right?
Yeah.
So, I mean, that should mean that, go to blue, okay. That should mean that positive ion should just go inside to make the inside more positive until there's a 0, +0 on the outside and -0 on the inside. Right? Diffusion states that should happen. Why?
Yeah.
Why isn't that the case then?
But nerves don't move via passive transport.
Well, everything that has any type of membrane between it will always have some type of diffusion will always be preferred because it doesn't take energy for it, right?
Yeah.
Yet nerve cell, that's why for a basic cell, right? Not a nerve cell, the inside is 0 and the outside is 0, right?
Yeah.
And if it wasn't, then, if the outside was positive and the inside was negative, positive stuff should go from the outside to the inside. If the inside was positive and the outside was negative, positive stuff should go from the inside to the outside. It should do whatever it takes to establish equilibrium.
Okay.
So why?
So that's what nerve cells do?
So nerve cells. So I'm just trying to show that in everything tries to reach equilibrium, right?
Yeah.
Yet in nerve cells, there's this weird negative, nerve cell. Why?
Mm-hmm.
Why? Why does it stay at the negative?
Well, because that's how it can maintain the equilibrium, no?
I mean, equilibrium would mean that the charge on the inside is equal to the charge on the outside.
Yeah. Well, isn't that what you drew there?
Well, here?
Yeah.
That's what it is for regular cells, but nerve cells, the inside is negative 70.
Yeah, but the outside, you said it's negative 72. No?
Well, the outside is like positive 70.
I'm positive 70. Yeah.
But that, I mean, how about this? Let's just say that there's one positive ion on the outside, one negative ion on the inside, right?
Mm-hmm.
So, what should happen, right? Wait, sorry, how about this? 2 positive on the outside, 2 negative on the inside, right? So, what should happen is this guy goes in and this guy comes out to give you 1 negative, 1 positive on the inside, 1 positive, 1 negative on the outside.
Okay.
So you go from 2 plus and 2 minus on the inside to
1 plus and 1 minus.
0 and 0.
Oh, because they cancel each other out.
Yeah. So why doesn't that happen here?
In nerves?
Yep.
Uh,
So here a concentration, voltage, whatever, gradient exists, right?
Mm-hmm.
And what is this type of transport? Passive or active?
That would be active.
Well,
Oh wait, wrong. Sorry. Passive.
okay. And passive does what to the gradient?
It dissipates the gradient.
Exactly, right? So, in order for it to go this way is passive, but to go this way, it's active, right?
Yeah.
So, what happens in nerve cells to maintain this concentration gradient is that active transport is done. So, passive...
Yeah, active transport would continuously need to be happening, so
Yeah.
that's why it needs energy on each side, because it needs to build the gradient each way.
Exactly. So basically, passive dissipates the gradient, makes it equal on both sides, and active creates the gradient. And for nerve cells, that's like the leak channels and the ATPase. So I just wanted you to understand completely. Like, passive carriers dissipate the gradient, active transporters build the gradient. And I'll just ask you one more, and then we can be done. Oh, perfect. This one right here.
Which protein is responsible for time D. Wrong.
Well, wait. It's either D or the leaky channels, but I forgot exactly what the leaky channels were. Let me see.
Do you have the?
Oh no, you're right. Good.
Yes, let's go.
Okay. All right. So okay, we'll pick up on this tomorrow. This chapter 15 PowerPoint. There's gonna be a lot of crap here. I've never seen.
Yeah, I know.
I've never seen so much about the Golgi, but yeah, we can do it. All right.
It'll be okay.
Yep.
Yeah.
All right.
We'll be okay.
All right. Cool. All right. Glad things are getting better for you health-wise, at least in terms of figuring it out.
In terms of finding out, but I haven't had anything better
yeah.
health-wise in pain.
Yeah. Sorry.
I still got a little bit of pain until then.
Mm-hmm. Yeah. All right. I'll see you tomorrow then. Okay.
See you tomorrow at 12.
All right. Take care.
All right. Take care.